So this is the second half of a lecture on selective and differential media for the week of April 5th, 2020. I already talked about in part one what selective and differential media are, and so I just wanted to remind you that they're mainly used to identify bacteria based on the biochemical properties of different groups, and differential and selective media can be either one or the other, so they can be just selective, just differential, or they can be both. And all of the examples in this particular lecture are selective and differential. And so the first type of media that I'm going to talk about in this lecture is EMB, or Eosin methylene blue agar. You can see it has this kind of nice dark bluish purple color when it's uninoculated. And EMB is selective and inhibits the growth of gram-positive bacteria, selecting for gram-negatives. And EMB is also differential. And the main thing that EMB can differentiate between are microbes that can ferment lactose or lac-positive microorganisms and microbes that can't, or lac-negatives. And you can see here lactose-negative microorganism growing on the far left, a lactose-positive growing in the middle, and then another lactose-positive growing on the far right. And the way that EMB differentiates between lac-positive and lac-negative, or lactose fermenters and non-lactose fermenters, is basically by the color of the colonies. And so strong lactose fermenters, or strong lac-positive organisms, will turn purple, as you can see up here, even all the way up into a dark black when grown on EMB. Microbes that can sort of or weakly ferment lactose will appear as a pink color, as you can see down here on the right. And non-lactose fermenters will have no color at all, as you can see up here in the top right. And so EMB is selective for gram-negative bacteria, and then differential for lactose fermentation. Another type of selective and differential media is MacConkey agar, and so you can see an uninoculated plate up here. It kind of has this light pinkish purple color, and MacConkey contains bile salts and crystal violet that actually inhibit the growth of gram-positive organisms and select for gram-negatives. And so you won't see any gram-positives growing in MacConkey agar. And MacConkey can also differentiate, once again, between lactose fermenters or lac-positive organisms and lactose non-fermenters or lac-negatives. And the way that MacConkey does this is using a pH indicator called neutral red and the sugar called lactose. And so by incorporating lactose and neutral red into the media, you give a lactose fermenter the potential to ferment lactose. When that happens, the pH of the media will drop because it produces lactic acid, and neutral red will turn pink, or dark, kind of this dark pink red here on the left. When lactose is being fermented, the pH of the media is turning low, and so a lac-positive organism on MacConkey will have this red color, whereas a lac-negative is unable to ferment lactose. The pH of the media will go up, and increased pH in neutral red will show up as a yellow media. And so you can see a lac-positive on the left, lac-negative on the right, lac-positive being pink on MacConkeys and lac-negative being yellow. And just a reminder that all of the organisms that grow on MacConkeys will be gram-negative because it's selective against gram-positives. The next type of media that's both selective and differential that I'm going to talk about is Mannitol salt agar, or MSA. And unlike MacConkey and EMB, MSA inhibits the growth of gram-negative bacteria and selects for gram-positives. And it does this by containing a very high amount of salt, or NaCl, so 7.5 to 10%, which is a pretty high concentration of salt, will inhibit growth of gram-negatives and not gram-positives. So that thick peptidoglycan layer can provide some protection for the gram-positives against this high salt, which is why they can grow fine on a plate like you can see up here on the top right. So this is an uninoculated plate. And once you inoculate an MSA plate with your organism, it can be used to differentiate between organisms that ferment mannitol, which is another type of sugar, and those that don't. Right? And so if you put a gram-negative bacterium on MSA, you won't see any growth because it selects against them, but gram-positives will grow. And then the addition of this mannitol sugar, as well as a pH indicator called phenol red, can help you differentiate between whether your particular bacteria can ferment mannitol or not. And so phenol red turns yellow at a low pH, and when mannitol is being fermented, it produces an acid that lowers the pH of the medium and therefore turns the whole thing yellow. And so a mannitol positive bacteria or mannitol fermenter will turn yellow on MSA, and the opposite is true for a mannitol non-fermenter. And so you can see on the right here a mannitol negative result where this bacterium cannot ferment mannitol. The pH of the media will actually increase, and that phenol red will turn a deeper, darker pink color. And so mannitol positive on MSA is yellow, and mannitol negative is kind of a dark pink or red. Another type of selective and differential media is bile esculin agar, or BEA, which is selective against gram-positive bacteria. It contains a high amount of bile salts that allow gram-negatives to grow really well and inhibits growth of gram-positives. And an uninoculated plate can be seen up here, and it has kind of a typical like beige-ish color. But BEA can be used to differentiate between two specific types of bacteria, the Streptococcus bacteria and Enterococcus bacteria. Because Enterococci are able to hydrolyze esculin, and esculin is incorporated into this media along with another compound called ferric citrate. And the hydrolysis product of esculin reacts with ferric citrate to turn the medium black. And so you can use a BEA plate and look for color change from beige to black to differentiate between an Enterococcus, which would have a black or dark brown media, and a Streptococcus, which will remain beige because it's unable to hydrolyze esculin and therefore unable to turn the media black. Hektoen agar is another selective and differential media that has a really pretty color, kind of a gusher's blue, if you will. And Hektoen is selective with the addition of all these dyes as well as salt against most gram-positive bacteria. And so gram-negatives will grow fine on Hektoen agar, whereas gram-positives won't. And so the uninoculated plate can be seen up here. It's got this nice deep teal, and once you plate organisms on it, you can use Hektoen agar to differentiate between lactose fermenters and lactose non-fermenters, or lac-positive and lac-negative bacteria. And so a lac-negative bacteria that can't ferment lactose won't change the color of the media at all, whereas a lactose fermenter will change the color of the media from deep blue to orange. There is a pH indicator called acid fusion in Hektoen media, and acid fusion turns yellow to salmon at a low pH. So when lactose is being fermented, the media is getting more acidic, the pH is getting lower, and it turns this orange color. And so your lac-positives will look orange, whereas lac-negative bacteria will remain, the media will remain blue. And Hektoen is interesting because it can also differentiate between bacteria that can produce hydrogen sulfide and ones that can't. And so in addition to these dyes as well as bile salts, Hektoen contains sodium thiosulfate and ferric citrate. And these compounds can interact with hydrogen sulfate and produce a black precipitate in the same way that TSI agar can. And so what you'll notice is that when a bacterium can produce hydrogen sulfide, the colonies themselves are filled with this black precipitate, And that's how you distinguish between a hydrogen sulfide positive bacteria, which has black colonies, and one that is hydrogen sulfide negative, which would have like greenish or bluish colonies. And so Hektoen is interesting because it can differentiate not only between different lactose fermenters, but also different hydrogen sulfide producers.